Dear students, please note down. These notes are available in the description of video. Please download your notes. This one is the fourth lecture of Nucleus. In Nucleus, we've already studied this outer nuclear coat. This is nuclear envelope. It is karyotheca. And this nuclear coat, which is double membranous coat, its outer membrane having ATS ribosomes. And this inner nuclear membrane, which is smooth without any ribosomes. But this nuclear coat is not continuous as it possesses nuclear pores. In our last lectures, we have also studied these nuclear pore complex because this nuclear pore generally consists of a cylindrical structure, that is annulus. Annulus plus nuclear pore collectively constitute a complex, that is nuclear pore complex. As well as we have already studied nucleolus. In today's lecture, we will cover this thread-like network. In this nucleus, it is chromatin. It is chromatin network, or it is chromatin reticulum. And this fluid sap part of nucleus, this semi-fluid substance of this nucleus, that is karyolymph, that is nuclear sap, that is nucleoplasm. As well as in today's lecture, we will clear just below this nuclear envelope, a fibrillar network is there. It is nuclear matrix or nuclear lamina. First of all, we will discuss the chromatin. It is hereditary material, and this chromatin, which is a DNA protein fibrillar structure, and this chromatin which produces a network inside this nucleus. That network is chromatin reticulum. This chromatin, thread-like structures, is differentiated into two parts. Very important for BSc students, BSc first year students, first semester students, final exam topic, that is chromatin. And this chromatin is differentiated into two forms. One is euchromatin, and second, this darkly stained structure, it is heterochromatin. Note down, in today's lecture, we will clear chromatin, this thread-like structure, which is made up of DNA protein fibrils. And this DNA protein fibrous structure, which is chromatin, differentiated into two parts, euchromatin and heterochromatin. Dear students, euchromatin means it is true chromatin. And this euchromatin, which is lightly stained, it is fibrous structure that consists of active genes. It is functional chromatin. And this heterochromatin, which is darkly stained, transcriptionally inactive. And this heterochromatin is further divided into two forms. It's two types, heterochromatin, two types. One is facultative heterochromatin and second type is constitutive heterochromatin. Dear students, this entire network which is present inside the nucleus, it is chromatin reticulum. It is chromatin network, that is a hereditary material which is made up of DNA protein fibers. And this chromatin having two forms, one is euchromatin, second is heterochromatin. This lightly stained fibrous structure, it is euchromatin, which is true chromatin having functional, having active genes. But this darkly stained, this region of chromatin, it is heterochromatin having inactive genes. And this heterochromatin having two types. One is facultative, second is constitutive. Now down, heterochromatin further divided into two forms. One is facultative. Facultative means it undergoes changes, means it represents temporarily inactivated chromatin. Dear students, its genetic part remains inactive for a short period because at that time its products are not required. That's why this chromatin remains inactive for that period. Temporarily, it remains inactive. But in this constitutive heterochromatin, the genetic metal, the DNA, remains permanently inactive. Now down, heterochromatin, this condensed chromatin, darkly stained chromatin, which is transcriptionally inactive. Its two forms. One is facultative, means temporarily inactivated chromatin, and second is constitutive, which is permanently inactivated chromatin. Note down, very important question for NEET students. Facultative heterochromatin means temporarily inactive chromatin, as in case of in somatic body cells of mammals like human females, in which two sex chromosomes are there, but out of these two sex X chromosomes, one sex X chromosome becomes inactive. That is facultative heterochromatin. And that inactive sex X chromosome represents bar body. That represents sex chromatin or bar body. If a female having two sex X chromosome, out of these two sex X chromosome, one sex X chromosome becomes inactive, means one bar body. Suppose a female, any somatic cell in a female having three sex X chromosomes, means two X chromosomes become inactive, means out of three sex X chromosome, two bar bodies. If question comes in the form of a female having four sex chromosomes, then out of four, three sex X chromosomes become inactive. It means in a female having four sex X chromosomes, three bar bodies. If sex X chromosomes are 3, then 2 bar bodies. If a female somatic cells only 2 sex X chromosome, then 1 bar body. This is the difference between euchromatin and heterochromatin. Again, important question for BSc student. Now down, euchromatin, that is true chromatin having functional genes. And this euchromatin, as shown in this diagram, it is fibrous in nature. It is narrow part. Thickness is very less, about 10 to 30 nanometer thick. Diameter very less. But here in this heterochromatin, this condensed part, much thicker. Diameter is about 100 nanometer. Narrow part, that is euchromatin, and this thicker part, about 100 nanometer diameter. It is lightly stained. This euchromatin, it is lightly stained, but this heterochromatin, it is darkly stained. Note down, euchromatin. Euchromatin occurs in the form of fibers, fibrous nature. But this heterochromatin, in which DNA remains inactive, condensed form, so it is granular. Euchromatin is fibrous and heterochromatin is granular. it contains active Gs. In this euchromatin, the Gs remain inactive, but in this heterochromatin, the Gs remain inactive. It takes part. This euchromatin takes place in transcription. Means this euchromatin is transcriptionally active, but this heterochromatin, it remains transcriptionally inactive. Replication. Note down, in this euchromatin, DNA replication takes place at early stages of S phase of cell cycle. As we know, in cell cycle, there is S phase, which is synthetic phase, DNA synthesis, DNA replication takes place. But here in this euchromatin, DNA replication at early stages, while in this heterochromatin, the DNA replication occurs at late S phase. Note down each and every point for competitive exams. Here, this euchromatin, it is very sensitive and it remains affected by several factors like temperature, pH, hormones, age, sex. But this heterochromatin remains unaffected with these factors like temperature, like pH, like hormones. Crossing over. In coming lectures, I will discuss all these topics: transcription, DNA replication, and crossing over, linkage. Particularly in genetics, I will discuss all these topics. Crossing over. Crossing over is very common in this euchromatin, but in this heterochromatin, it inhibits crossing over. And this euchromatin that produce the maximum part of this network, while this heterochromatin, it produces very less part. Nucleoplasm, the fluid, the semi-fluid substance, the transparent fluid of this nucleus, that is nucleoplasm, nuclear sap or karyolip. And in this nucleoplasm, which contains nucleosides, which contains some enzymes like DNA polymerase, like RNA polymerase, like phosphorylase for the synthesis of DNA, for the synthesis of RNA, as well as for the synthesis of nucleotides. And the last part of this nucleus, that is nuclear matrix. Note down, this nuclear matrix just below this nuclear envelope, it produces a thick fibrous network. And in this nuclear lamina or matrix, the terminal parts of these chromatin are embedded. It means this nuclear matrix or nuclear lamina, it functions as scaffold of chromatin. Means the terminal parts, the telomeres of chromatin remain embedded in this matrix. Dear students, this nuclear matrix provides mechanical strength to the nucleus, as well as this nuclear matrix, which is fibrillar in nature, like just below this nuclear cord, in which terminal part of chromatin remain embedded, involves in the formation of nuclear pore complex. This is all about nucleus. In this nucleus, we have covered all components: nuclear envelope, the nuclear pores, the nucleus, this entire network, thread-like network, reticulum, this one is chromatin, and this small little nucleus, that is nucleolus, the fluid of this nucleus, that is nuclear sap, nucleoplasm, karyolymph, and the last part, which lies just below the nuclear envelope, the fibrillar network, that is nuclear matrix or nuclear lamina, in which the terminal parts of these chromatin remain embedded.